And when we are thinking about life on Mars, there's two basic questions we can ask. One is, was there ever life on Mars? And the second would be, if so, is it still there? Okay. So we can essentially look for life currently on Mars. And if we never find life currently on Mars, does that mean there was never life on Mars? No. Okay. So we have to ask the other question, well, even though Mars is a crappy shithole of a planet now, and we maybe don't expect much in the way of living things on Mars today.
It's no South Dakota.
It's no South Dakota. We do know that Mars is different in the past, so those are two realistic and viable and different questions. Okay, so we're interested, is there still life on Mars? Because that would be way cool from a biological perspective. Why? I can tell you it would be way cool if there were life on Mars, but why would we really be interested in whether or not there's life on Mars?
Because if they can live there, maybe we can.
Okay, so a very pragmatic kind of...
I was just going to say, there's always been this question of, are we the only ones?
Are we alone in the universe? One of the fundamental questions, right? So if we find life on Mars, then what does that say about whether we're alone in the universe? We're not. Does it matter? I mean, we're not going to find little green men. We're not going to find intelligent aliens. We're not going to find an advanced dying civilization on Mars. If we find life on Mars,
It'd be like microorganisms.
what we're most likely to find will be microorganisms. How excited are people going to, in our culture, going to be if we find microorganisms on Mars?
They're going to be really excited. They're going to cheer on NASA for...
It's going to be riots.
I feel just the average person view, when they think of life on Mars, they think of us. So they're microbes, whatever.
Yeah. Yeah, it'll be
You'll get a few likes on Facebook.
yeah, it'll be trending for a little bit, and then some conflict will take it off the front page.
And that opens up the gates to having life on other planets outside of our solar
Right.
system.
So even if there's microbial life on Mars, then does that indicate that, well, potentially maybe there is a lot of life in the universe?
Is it possible for them to accelerate the evolution of the microbes?
Can you stimulate mutations? Sure. But when you say accelerate the evolution, that has built into it or packed into the idea that evolution is this directed thing that has some progressive endpoint. And we know that evolution really doesn't work that way. Evolution is a much more of a undirected process. Sometimes lineages become more complicated and more complex over time. Other times, other lineages, in terms of adapting to their environment, can become less complex. So it's not like evolution has this goal that we can speed up or slow down. But we can certainly, if we find microbes, we could tinker with them and we could expose them to things that would potentially create mutations and see what impact that has. One question about if we find life on Mars is, it's very interesting if it is actually an independent branch of life, right? Because none of you are biologists, but biology on the Earth has one big limitation. We have essentially one data point. We know that all living things on the Earth essentially share common ancestry and therefore are drawing on the same kinds of biochemical systems and so forth. We don't know if life needs to be organized the way life is on Earth because we essentially have one example. So life on Mars, if we find it, might be a second example of life, which would double our data points, right? We'd have, even if they're little tiny microbes, we could take a look and, well, how do they handle energy metabolism? How do they handle reproduction of genetic information? Are they using the same systems or are they using different systems? We might find life on Mars and realize that it is actually not a second data point. Why?
Because it could be exactly the same, or it could have the same ancestor as we do.
How could life on Mars have the same ancestor as life on Earth?
Alien.
Those damn aliens spreading life around the universe.
Wait, if we pick up like microbes from on Mars, we are like the abducting aliens. That's so weird.
Yeah, so what about comets?
Comets would hit the planet and then it would spread.
Okay. Yeah. So we could find out that we have successfully found life on Mars and we're jumping up and down, and then we realize, oh, well, it's still cool, but it's actually just another branch of us. Or we are a branch of them. We know Mars and Earth have been spitting rocks back and forth at each other for 4 billion years. Impacts on Mars blast material up that lands on the Earth. Impacts on Earth does the same thing to Mars. It's actually a little bit easier to hitchhike from Mars to Earth. So if we are, if we do have common ancestry with Martian life, it's possible that, that life could have gotten started on Mars first and hitched a ride to Earth. Because there's also some indication that, Mars might have been habitable earlier than the Earth. So yeah, all sorts of interesting reasons why we might be interested in life. Our first foray really into this question were the biology experiments on Viking. And, these were the Viking biology experiments. Which of those two life questions are they asking? Were they asking, is there life on Mars or was there life on Mars?
I actually didn't listen. I didn't hear you at first. I think they were just looking to see what was there.
Yeah. So this is these were the first landers that we sent to Mars, and right off the bat we tried to see, can we find evidence for current life on Mars? Okay. So yeah. So this is asking the question: is there life on Mars? How do you look for life on Mars or another planet? How do you identify life? Respiration. How do you define life? What does it mean to be alive?
To use energy.
Okay. So everything that uses energy is alive?
No.
No. If you were going to look for life on Mars, what would you look for? Now, if there's a stegosaurus kind of creature lumbering across the landscape, and you see it with a camera, that's easy.
Yeah.
But that's not what's going to happen. So, what? Okay, so we know life on Earth is built around cells. So maybe we can look for cellular structures. That's really tough to do with a spacecraft. So what they focused instead on was, well, we know living organisms metabolize. You and I and all living things on Earth are essentially just very complex chemical systems that are metabolizing energy sources from the environment in order to maintain our complex structures. Because if I keeled over dead right now, what would happen to the structures in my body? It would shut down, and then they would, what did you say?
Break down.
It would break down. So complex structures naturally tend to break down over time. That's just entropy of the universe. You leave things out, and things get more and more progressively disordered. And that's not a comment on any of your rooms or whatever, but that's just... Organisms have to metabolize energy in order to maintain these complex structures that they are. Okay. And so the Viking biology experiments looked for different aspects of metabolism. Is there the equivalent of respiration where things are being broken down? That was the labeled release experiment. Are there, is there metabolism going on that is taking simple materials from the environment, like carbon dioxide, and using it to build up more complex structures? That was the pyrolytic release experiment. And then the gas exchange experiment basically just said, well, if we expose a sample of this soil to our testing conditions, is anything happening with gases given off or taken up? Okay. So you guys all went through this this simulation of the Viking biology experiment. What did you guys see when you did your experiments?
The results were inconclusive because they all won against each other.
Okay, so results were inconclusive, but what were some of the results you saw? Well, let's speed this process up. Here we've got the labeled release experiment. We've got the experimental samples on the left, the control samples on the right. Again, this is the experiment where we take food in terms of complex organic material that we're dumping into this test chamber. And that food has been labeled with radioactive carbon atoms. And this experiment tests to see whether any of that radioactive material gets released from the broth and releases carbon dioxide or carbon monoxide or some gas that's given off by essentially this would be the equivalent of respiration. So for the experimental treatments, you add the samples of soil at day 0 and then just measure the accumulation of radioactive carbon dioxide over time. And do you see a response?
Yes.
Things are being broken down. And so this radioactive carbon dioxide is accumulating. What happens to that accumulation over time? It slows, it plateaus off. Why would you expect that to happen?
They've run out of things to break down.
They've used up all the food. They've broken down all the food. They've converted all of that labeled nutrient broth into radioactive carbon dioxide gas. So this is exactly what you would expect if there were little critters, little bugs in the soil breaking down the food that we're providing. And the function for the control over here. Well, yeah, we're not seeing the same response in the controls, but why run the control? Why don't we just say, oh, look at that. We got the evidence we want.
Because if it did the same thing when there was no life, then it would be...
Right. So this control is supposed to equal no life, right? And that is set up by vigorously heating the sample to kill off all the soil. So in this case, does heat sterilization eliminate the response?
Yes.
Yes here, mostly yes here. But if you look real close, this is heating up the soil to a lower temperature, so it's not quite as strong as sterilization. So we add the soil that's not been sterilized, and we see a response, and we sterilize the soil, and that response goes away. What is our interpretation?
That there's life.
We have, this is exactly what we would predict if there were life in the soil. So you can imagine there was a little bit of excitement at Mission Control when these results came down. When I did this simulation last year, at least one of the groups during the simulation asked me, well, are you giving us the real Viking data? I said, yeah. Well, it can't be because we're finding evidence for life. And, yeah, this is evidence for life. None of the groups were as excited this semester, so I'm a little bit bummed, but anyway. We'll go through the others in detail, but with the pyrolytic release experiment, we find that materials are being built up. Which again is what you would expect if there's life. But in this case, the control samples, the samples that were heat sterilized, also had the accumulation of carbon. So that's where it starts to get messy. And in the gas exchange experiment, there were lots of changes in the gas composition following addition of the Martian soil. One of the big things I want to highlight is that for many of the samples that were tested, there's just a whole lot of oxygen gas given off as the experiment was carried out. This result here is for the gas chromatograph mass spectrometer. And what is it showing? The GCMS was looking for what? It was not actually part of the biology package. But just in general, what was it doing?
Just looking for gas exchange.
No, that would have been this one up here. The GCMS was checking the soil for what? What kind of material was it looking for in the soil? Organic material. And so they saw these results, and the interpretation is we're not seeing any evidence really for any organic material in this Martian soil sample. So the labeled release experiment is spot on results, what you would expect if there were life in the soil. But we find no organic material in the soil. And the other two experiments are showing some reactions, but the controls are showing the same reactions as the experimental. So if those reactions in the gas exchange and the pyrolytic release experiment were due to living organisms, you would think that sterilizing the soil would eliminate those responses, and they didn't. So, what's your overall interpretation?
There's no life on Mars.
There's no life on Mars.
We don't know
Currently. We don't know. Anybody else? It's hard to tell. Results are hazy. It's like the magic 8-ball. Okay. So, the more results came down, the more confused the situation became. The fact that there was no evidence for organic material in the soil really turned most of the scientists off from the idea that these results were due to living organisms, because we can't imagine putting together living microbes without, building up some organic material.
I mean, isn't that only thinking of the way that we develop?
This is life as we know it.
Yeah, but I mean,
Right.
it's a bad way to look at other.
So, yeah, how do you go about looking for life not as we know it?
I don't know.
We don't know.
We don't know.
Look
Okay.
at every molecule underneath a microscope.
Yeah. So, I mean, clearly this was a very ambitious first test for life. I mean, we had no previous even chemical data on the soils of Mars. And we go to try to do home run, touchdown, whatever on the first mission we send. These results are still controversial, as I'll show you in just a minute, and this will probably be what we finish off with today. But the majority of the researchers looking at this data come up with the conclusion that there's something interesting going on with the labeled release experiment, but we have no evidence. We cannot make the claim from what we have collected here that that's due to life. So there are some interesting responses. But most of the scientists involved suggested that it is not life that was responsible for these responses, but more some kind of exotic chemistry that is not life. And what people, what a number of researchers suggested is that given that Mars has no magnetic field and very little atmosphere, and the soils are exposed to the solar wind constantly, that there is just a lot of oxidizing material in the soils, like bleach. Think of it as bleach. You put bleach in and it oxidizes stuff and breaks it down and so forth. So, that response by the labeled release experiment was thought to be, well, we're just putting the nutrient broth in with this soil that's got all these oxidizers in it, and that's breaking down the nutrient broth and releasing the radioactive carbon dioxide. And if you heat up that soil, it breaks down the oxidizers, and therefore you're killing off the reaction, but that is not necessarily indication that there was life there. And that was the interpretation for quite some time. But more recently, people have been re-examining the Viking results. And frankly, the scientist who was responsible for the labeled release experiment, to this day, he continues to argue that no, there may have been problems with the other experiments, but his experiment found evidence for life. But other people have come in and reinterpreted the results as well. Let me simplify this a bit. A couple of these researchers, this guy in particular has done a lot in the field of searching for life elsewhere in the universe, not just on Mars, but other places as well. And this is somewhat one of the life not as we know it kinds of suggestions. So life not as we know it. These two researchers, on the basis of just understanding chemistry and organic chemistry and what kinds of situations might be present on Mars, have suggested that we could have Martian life forms that are water-based, but not just solely water-based, water and hydrogen peroxide-based. The hydrogen peroxide could act as a way of maintaining metabolism at low temperatures. And so, this kind of Martian metabolism, or potential Martian metabolism, that's incorporates this action of hydrogen peroxide. How many of you are familiar with hydrogen peroxide? What do you use it for?
Disinfectant.
It's a disinfectant. You pour it on your wounds, and it oxidizes the bacteria. But you could imagine bacterial metabolism that would actually incorporate hydrogen peroxide into the metabolic processes, and that presence of that hydrogen peroxide would make those living organisms stable at a much lower temperature than we find here on the Earth. So this is a reasonable kind of life, not as we know it, that might be suited for Mars. This was just a theoretical model, a theoretical exploration of what could be possible. Again, let's just reiterate some of the aspects here. This water hydrogen peroxide mixture, as I said, reduces the freezing point, makes it easier for these Martian microbes to deal. Potentially would make it easier for these Martian microbes to live under these Martian conditions. If you took these kinds of organisms and dumped them into the gas exchange system that we sent to Mars in the Viking mission, these critters would die, they would break down and would give off tons of oxygen as they did so. In fact, it's possible that if these kinds of organisms were actually living in Martian soil, they would be killed off by the experimental conditions that we sent on the Viking mission to look for life. Basically, the fact that we're dumping this nutrient broth on is very much un-Martian conditions, and that excess of water actually would be lethal for this kind of metabolism. So it just, again, reinforces the complexity of how do you actually design a set of experiments to go look for life on Mars when you haven't even dealt with some of these questions of, life might not be the same as we find it here on the Earth. But again, this is all just what?
That's awesome.
That's awesome, right? Okay. So, but this is again just kind of a theoretical model based on what we know about how chemistry works. This kind of metabolism would be well suited for Mars. It's not actually evidence that there is that kind of metabolism, those kind of critters on Mars, but it again kind of just brings into question the negative interpretation of the Viking results. Might not be so negative at all, after all. What?
How recent is this?
This is maybe 5 or 6 years old. Okay. So, I mean, this is several decades after the Viking missions, the hooplas, all died down. Yeah. Yeah, I won't go through any of that. Okay, so this hypothesis actually explains a lot of the Viking results. The GCMS did not see any organic material. Well, duh, when you put this kind of life into the GCMS we sent, all of that organic material is going to be broken down by the hydrogen peroxide that is part and parcel of this metabolic system. The release of oxygen, the liquid water being lethal would, kill off the life that we're looking for, and so forth. So again, it just reinforces the point that we cannot necessarily say that because the results of the Viking mission were messy, we have evidence that there is no life on Mars, especially life not as we know it. Well, yeah, at some point we will feel like we're confident enough to go back and actually look for life on Mars. We've been following the water, and now Curiosity is looking for habitable environments. And the European Space Agency, on their upcoming lander mission, is sending along some life detection experiments. And they're not going to be foolproof, but they're going to be different than what we sent with Viking. They're going to be looking for organic materials in a way that would not break down those organic materials, if this is what's going on. They're going to be sending instruments that will look for, our amino acids and sugars that we use in our bodies are all, right-handed or left-handed. If you have a complex chemical, they can exist in mirror images, and biology tends to focus on one type of chemical versus the other. The Phoenix lander, which we talked about in terms of water, so I don't need to go through all of this stuff again. It basically sent a chemistry lab to Mars, so we're actually starting to get some of the idea of, well, what is the inorganic chemistry of the soil like? And again, this perchlorate that was found that might have implications for water and brine and so forth could also have implications for Martian life. As I mentioned, there are some organisms that use perchlorate as an energy source or as part of their metabolism. And the perchlorate, you can build the same kind of case around a water perchlorate kind of metabolism that you can with the water hydrogen peroxide. Has the same kind of effects in terms of reducing the freezing point, stabilizing life at low temperatures and so forth. So other people have reanalyzed the Viking results now that we know that there are all of these perchlorates in the soil of Mars. And the perchlorates themselves would explain why there was no organic matter found in the GCMS experiment on Viking. Again, you take a soil sample, you begin to heat it up. The perchlorates in the sample that we know are there now because we've actually done some chemistry would break down the organic matter before it had a chance to be sampled by the mass spectrometer. So in one of the more colorful comments about the Viking GCMS experiment, some of these researchers have taken soil samples from some of the dry desert areas in Chile and run them through an equivalent GCMS as what was on Viking, and found essentially the same no level of organic matter. And I'll just leave it at that. So again, if you think back to 1976, the fact that the results coming down seemed to indicate there was no organic matter in the soil really somewhat took biology off the table for most of the researchers. Those GCMS results in particular are the ones that are being most questioned, given now that we have a better understanding of what's going on in the soil of Mars. And again, these guys who originally put together the water hydrogen peroxide model have come in with another model. This is about 5 years old now. Basically saying you can do the same thing with perchlorates that we did with, theoretically with hydrogen peroxide before, that you could have a life form that would use the perchlorates that are naturally occurring in the Martian soil as a fundamental part of their metabolism to stabilize their metabolism under these cold temperatures. So, and this is just, the recent Curiosity mission in the results that they recently released about there being the potential for liquid brines in the environment. This is also related to this question of perchlorates. So, kind of a weird molecule that you don't encounter much in day-to-day life unless you do like model rocketry or various industrial processes. But the fact that we now know that perchlorates are such an important part of the Martian soil gives us a different way of thinking about how we might look for life on Mars that hopefully will be a little bit more sophisticated the next time we go about it.